Hi, in this video I'd like to briefly go over HPV cancer and how exactly this HPV virus can lead to cancer formation in humans. So the majority of the information and data in this video is originating from this article here published by Claudia and Junji from the University of Texas. And this is a recently published science article on the biological and clinical aspects, but we will be focusing on how exactly normal human cells, after being infected with HPV, especially the ones that are high risk HPV, strains 16 and 18, that lead to cancers. So almost all cervical cancers are HPV associated, and almost all of them are associated with high risk HPVs. So specifically HPV 16 and 18 are the most deadliest. They are the most known to cause cervical cancer, but they are not as common. But if you are positive with these two, you have a very high risk of cervical cancer. And then there's also an increasing number of head and neck cancers, especially oropharyngeal cancer, which means the cancers of the respiratory tract, and also anogenital cancers, which means anogenital means includes the as you can see here, it includes the vagina, the penis, the vulva, and also the anus, basically the entire area of your lower body, or in the region of your genitals and also the anus. So testing for cervical cancer has improved a lot because of the discovery of the vaccine. The vaccines are specifically effective against strand HPV 6, 11, 16, and 18. And that's why for cervical cancer, testing and preventing has improved a lot. But for those that cause anogenital cancer, it is still very much still a problem. So research is still required. So in this slide here, we need to understand what exactly is a cell cycle. So the cell cycle, generally, it is divided into 4 phases, as you can see in this image here. So first phase, we'll be calling it as G1 phase or growth phase 1. Second one is S phase, which is also known as synthesis phase, and then G2 phase, which is growth phase 2. Then into cell division, the mitosis cell division, where one cell becomes 2 cells. So G1 phase is generally where the cell spends most of its lifetime, where it grows in volume, in its organelles, and repairing any damage. This is where it's going to spend 90% of its life in this stage. The other 2 stages are in preparation for cell division. So S phase is a synthesis phase where it replicates the DNA, where 23 chromosomes now become 46 chromosomes in preparation for the upcoming cell division. There are also other processes involved, but most of it is geared towards preparing for cell division. The growth phase 2 prepares certain organelles, such as the microtubules that are involved in cell division, mitosis, and also it prepares mostly to enter into the cell division stage. And cell division, once it begins, this is where one cell divides into 2. So this is, in general, basic understanding of the cell cycle. So we understand that according to this article, we know that high-risk HPV strains, they infect human cells by generating specific oncoproteins. Now, in this article, it states here, it says E6 and E7. So these 2 are the viral oncoproteins of question, and they lead to the degradation of, respectively, P53 and PRB. So P53 and PRB are simply tumor suppressors, tumor regulator genes. They are very important genes that are found inside the cells. Almost all cells have these to help regulate and control the cell cycle and also prevent the formation of a tumor. So what E6 and E7 does is that they affect the function of these 2 very important suppressors and regulators, thus leading to the formation of the tumors and cancers. So it also states here that P53 is a gene that makes proteins inside the nucleus of cells It plays a key role for controlling cell division and cell death. So basically, p53 is involved in apoptosis. It activates apoptosis when it detects that a cell is damaged, has some irreversible DNA damage, and should not be allowed to replicate. And this is why p53 will be activated, causing cell death. Prevent that damaged DNA damage or infected cell to enter into the cell division stage, preventing it to spread. PRD is a key regulator of the cell cycle. It uses its growth suppressive effects to prevent excessive cell growth. Yes. So PRB is what lets G1 phase to enter into the S phase when it is inactivated. So an activated stage will lead to cell to divide and grow. So in this here, I'll move it here. Okay, so in this image here, you can see the pathway of how E7 and E6 lead to the formation of cancer cells, how your cells turn into tumors. So E7 inhibits PRB specifically, and E6 inhibits P53. They are very specific. So as we mentioned before, PRB is involved in apoptosis. So once a cell is infected with HPV infection, especially the high-risk strains, the PRB will be degraded when E7 and PRB bonds together. So there is no longer a PRB present in these cells. So when these cells experience DNA damage or radiation or chemical exposure that damages the cell, PRB is what initiates apoptosis. But since there is no longer a PRB present due to degradation, the cell continues to divide because there is no longer a PRB controlling the cell cycle. So in this case, we call this that the cell has a deactivated or inactivated apoptosis. So as for E6, E6 blocks apoptosis by causing P53 degradation. It causes the cell to continuously grow and proliferate. It arrests the cell in the G1 stage. So E6, when it binds with the P53, it will degrade P53. And once P53 is no longer present, the cell continues to stay in G1 stage and grows continuously, leading to tumors. So in this slide here, you can see some general stats that I got from the paper. We can take a look one by one. So according to this paper, the most prominent forms of cancer is lung and bronchus cancer, female breast cancer, colon and rectum cancers, and then the rest are not as common. So as you can see, it shows incidence rate and also mortality rate. This is something that you can see. And what you will notice is that female breast cancers have very high incidence rate but very low mortality because there's a lot of prevention and the screening tests available nowadays, so that it's very easily preventable as long as you detect it early. Some cancers do not share the same kind of mortality because it might be very difficult to screen or detect this type of cancer, and usually when it is detected, it's already at a very advanced stage. So this is why we need to close the gap in research to improve healthcare. Next one is localized in the US, as in the most common cancers and also its mortality, and it's very similar to what happens in the world. This is a more informative chart where it shows only cancers between males and females. What is the most common one? So for males, the most common one is a prostate cancer. As you can see, it is without a doubt the most common occurring cancer in males, and in females, breast cancer is the counterpart, as in the most common cancer in females. And then the next one is lung cancer, and females is also lung cancer. And the rest seems to be quite similar. These are something that we can take a look. I'll share the link of this article down in the description box if you want to take a look. So uh, in this chart, we can see the cancer incidence rate in the US with a few factors here, the comparison of females and males. So tobacco use is one of the most prominent factors because it will suppress your immune system and also make you a lot more vulnerable to certain cancers, infection. Obesity is definitely a factor. If you are unhealthy with a very weak cardiovascular system, you may be at high risk. Next one is alcohol use, physical inactivity, a very crucial one, and also lastly, HPV infection is also a factor in getting cancers. You can take a look at uh, cancers that are caused by viral infections. So not all cancers are viral infections, but there are uh, uh, there are a few that are caused by viral infections, which means infection caused by viruses that lead to cancer. So in this chart here, we can see that the most prominent one is not HPV, but instead uh, Helicobacter pylori, which is the most common one. And the next one is HPV. And then following two are hepatitis B virus, hepatitis C virus, and then there are still a small group of unknown that we suspect it to be caused by viral infections. Coming to the chart on females, the most common cancers that you might uh, contract after being infected with high-risk HPVs. So as we said before, HPV 16 and 18 are the most high risks, and they have a very high risk of causing cervical cancer, up to 7 to 8,000 females per year once you're infected with 16 and 18. And they also lead to other forms of cancer, oropharynx, which means the respiratory tract and the oral cavity, the vagina, the vulva, and also the anus. But without a doubt, cervical cancer is the most prominent one. Other forms of HPV strains, such as the one listed here, 31, 33, 45, 52, 58, also have a risk of causing the similar cancers, but significantly lower. So these types of strains, we will know them as low risk HPV. And then the remaining of other HPV infections, which have very low risk of causing any form of cancer, and usually will be dealt with your own immune system within a year or two. In males, 16 and 18 will not cause you cervical cancer, but you have an entirely different problem. It is by far the highest in causing oropharynx cancers, which means cancers of the respiratory tract, the throat, and also the oral cavity, which we can see in this chart here is up to 8,000 to 9,000 cases. Highest risk in males leading to oropharynx cancer. It can also lead to anal cancer or penile cancer, but as the chart suggests, you have a significantly higher risk of contracting oropharynx cancer. And the other low risks strains, 31, 33, 45, 52, 58, other strains also may cause these cancers in men, generally three, penile, oropharynx, or anal. But oropharynx cancer is the most common one that we see in males. So these two pie charts are reflecting of these two bar charts. So this is a recap on how E6 and E7 infected cells lead to tumor formation, and also E-regulation of the cell growth. So E6 is specific to P53 and E7 is specific to PRB. Once P53 is degraded, it will cause the cell to be in continuous growth, leading to this tumor formation. And in E7, specific to PRB, apoptosis will be inactivated, causing the damaged or infected cell to proliferate and grow, leading to cancer. Finally, I'd like to mention ongoing clinical trials that are currently underway for uh, treating HPV cancers. Because at the moment, there is no single form of highly effective form of treatment that can treat cervical cancer, penile cancer, and anogenital cancers. But research is underway. The article mentions immunotherapy, which means using monoclonal engineered T cells and antibodies as substances mimicking the compounds of the immune system. So in many cases, people who develop these types of HPV-related cancers are immunosuppressed, which means their immune systems are not functioning well. Their T cells and B cells are weakened and do not carry out their function. So these type of people will need an immuno boost. These types of clinical trials are focusing on boosting the patient's immune system to help them overcome this cancer treatment. And then currently for HPV cancer, cervical and head and neck cancers, new form of immunotherapy using anti-CTLA4 monoclonal antibody is currently in phase 2, which means it's quite prominent, quite promising. This ipilimumab antibody. It is important to note that these types of clinical trials are either administered alone with the immunotherapy or in combination with radiotherapy, because cancer cells do not respond well with radiotherapy. But its side effects are that normal human cells also are affected.